Hello, everybody. Welcome to today's session on butterfly experiential walkthrough with Dr. Krushnamegh Kunte. So we all know Krushnamegh is an eminent butterfly biologist of India as well as the world. So today he'll be here telling you guys the correct way to take the point count, so that you guys can take your counts and share it with the BBM email. And lastly, I would like to thank him from NatureMates because he has been our mentor for so long on our various butterfly projects. So really happy to have you here today, Krushnamegh, and the session is yours now.
All right. Good morning, everyone. It's a pleasure to be here. Following the chat yesterday when we talked about 30-minute counts. This is a method that we have been using to count butterflies in India for the last 12 or 13 years, including almost a 10-year data set on butterflies of Bengaluru. So what we are going to do today is we are going to walk in this area, and I'm going to demonstrate how we do this, how we typically have been doing it. So first of all, you start with whatever small group you have. In this case, we have 5 of us, and we are going to walk along this path. And will actually do a 30-minute count here, and then you will see how we normally conduct it. And after that, if you have any questions, we can quickly address those. After we will shorten it a little bit just for the demonstration purpose. So what we do is, first of all, we start a file. Here, I'm going to record data on my mobile phone. Eventually, our app will be released. So when the app is released, you can also do it directly on the app. But you have to start recording all the information that may be relevant to the 30-minute talk. So what are the things that you should absolutely record for every single talk? So if you're going to do this on a particular day, you have to start with date. So today is 12th of September. So I first mentioned it's 12th September 2021. And then there's Krushnamegh Kunte, Viraj Nauge here, Sarika Vaidya, Archan Paul, and Arko. So there are 5 of us. So I've mentioned names of all these people so that we have a record of exactly who was on the trail. And then this is near NCBS campus, GKVK, wooded area with agricultural fields. Can you just move around to show what kind of area this is? So this is a nice road with some woodlands and then some agricultural fields here. So this is where we are going to take the count. And then today is unfortunately quite clouded and breezy. There's very light sunshine, but otherwise it's not exactly a very good day to watch butterflies. Butterflies really like it when it's hot and humid. That's the time when they're most active. So again, choose your days carefully. If you're constrained by when you can do the walk, for example, you can do it only on weekends, then sure, you will have to just do it when you can, whether the weather is good or not. But otherwise, if you can choose when you would like to do butterfly walks, then maybe look at the weather forecast in advance, and then of course choose your day and time carefully. So I've recorded all these things, where we are, who's in the group, date, and then of course that it's clouded and breezy. Also the habitat type. I would ideally also take a few pictures along the way. Today we're going to record, so we'll actually get a video recording of the entire habitat. But if you're in a fairly large group, one of the things you can do is once in a while, once in 2 months, 3 months, something like that, you can also go around your path, butterfly count path, and take a video perhaps of the entire thing so that you can record of what the habitat looks like. So that's one thing that you could do. The other thing that you could do is pictures at some regular intervals. AM start after that you start your actual count. So it's 10:40 now. So you'll start walking. So walk at a normal pace, walk at a pace that you're comfortable with, and um, watch butterflies. Now, if the density of butterflies is really high, then you're going to walk really slowly. You're going to stop in a lot of places. You're going to spend some time identifying butterflies, noting them down, and then you're going to start walking. Four people, something like that. If you have that kind of a group, then it's much easier to do this. But one person is not too bad. All right, so I see a common, small grass yellow there. So I'm going to record that. There are two individuals. So I've recorded two. Yes. And one Delius. Okay, so that is Indian Isabel. Okay. And what we should do is, if there are multiple people, ideally, if you are all compiling data, only one person can write down, and then other people can walk around a little bit, maybe take some bushes or keep looking around, keep looking up, down. There's one more genius there, but it's probably the same one that you saw earlier because it was in the same area. So this is what I described yesterday. If you're walking in a particular area and you see the same species there again, you can assume that it's the same individual unless you see two separately. Okay. So here she had seen it earlier and I just saw it. So we are just going to count it as one individual and then keep walking. So we have got three species now. One lemon immigrant that we just saw, and then two spotless grass willows earlier, one Indian visible. So this is okay. Now there is another Indian visible there. So that's the second one. So from one, I'm going to make the entry for Indian visible to be two individuals seen on this count so far. And if you see any marvelous plants, this is lantana, and there is another plant belonging to Fabaceae, on which a lot of lysinates feed and other things feed. There's ipomia here. So keep looking around. Don't just look for butterflies. Also look for caterpillars, eggs. If you find any of those, maybe you can even hang around. Perhaps females will come back and lay more eggs, or generally keep an open eye. Here you have Passiflora. I don't see any caterpillars here. Probably Tonicostas not breeding here. You could also even write down notes like this on the side. They won't be part of the 30-minute count data itself, but they will be. There's a line blue, most likely line blue. What you could do when you're not sure about the identification, you can just say Prosodus species or line blue species. And later on, if somebody is able to identify that butterfly because it's sitting, earlier it was flying, so you couldn't really identify it. But if it sits down after some time, then you can, of course, enter that information. But right now, I'm just going to say line blue species. And there's a single in there. Okay. If there are flowering plants, for example, there's some large tree there, which I don't know. It's like some kind of a yeah. And then you have another sapling daisy here. There's another invisible, and of course you have lantana. So if you have good patches, this is actually a very good patch of lantana. It's just not a small lantana, but actually a fairly extensive shrub. So when you find a good spot like this, you can also hang around for a little bit. That line blue is sitting down now, and it has a. Uh, I think it does not have a tail. So that will be a tailless line blue. So earlier we just saw it flying around. Now we see it sitting and it's identified. So I'm going to change that from line blue to tailless line. And of course, if you see more line blues on this tail, which you didn't see sitting down and therefore couldn't identify, then you can bring back line blue species in your data set, and that should be fine. Okay. So let's hang around here for a little bit since there is a good patch of lantana. Uh, and we'll see if any more butterflies come out. And look, there are two, three of us actively looking around, and we hadn't seen this earlier. And I just see there's a bush brown there. Yeah. Can you actually come close? Go close to that and take that bush brown. So it's sitting here. Yeah. So this is the bush brown, and from experience, we do know that in on this campus, we largely get dark brown bush brown. So for now, I'm going to write it as dark brown. Typically, if you have permission from the forest department to catch and release butterflies, what you could do is catch it, make sure that it's a dark brown bush brown by looking at the upper side, forewing, which distinguishes dark brown bush browns from others. Unfortunately, normally they don't sit with wings open. So most of the bush browns that you see, you're not going to be able to identify from this dark brown bush brown, common bush brown group. So in that case, you can either just say bush brown species, but here, for example, we know common bush brown is very rare and dark brown bush brown is the most common one. And the markings also look like that. So here, because of prior experience, I'm going to say dark brown bush brown. If you don't know, then you should just write bush brown species. But of course, we have permission to catch butterflies here in Karnataka. So if you want, we can catch it, confirm identification, and then release the butterfly here. Okay. So this is why you should really not walk very fast. You should really spend time in patches where you might see butterflies. And let's wait here for some more time and see if any more butterflies come around here. So again, keep looking and definitely no chatting with each other when you're doing counts unless everybody is still looking around. Because if you're chatting with each other, you're not actively watching butterflies. You can talk about butterflies, but don't chat generally. So there's one more Jezebel. But since we have recorded two Jezebels here, we don't know whether it's the same Jezebel going round and round. So in these cases, when you see butterflies and you keep seeing them around, you don't know whether it's the same one or different ones. What you could do is through your binoculars or with your camera, you can take a picture, see if there are any markings which help you distinguish one individual from another. Maybe most butterflies will suffer some wear and tear as they're flying around. They brush against vegetation. They are attacked by predators. They get caught in spider webs. All these things do some damage to the wings. And then that damage can be fairly peculiar so that you can distinguish one individual from another if you're seeing multiple individuals in an area like this. So in these cases, there may be two Jezebels, there may be five. Here you should really look for ways to distinguish between different individuals and then note those down. One skipper. Okay, so I'm going to write swift species because here you mostly have swifts. You also have brown owls and common banded owls. So there are few skippers, but what she just saw was a swift. So I'm going to write swift species. Now, unfortunately, swift is a group. It's an entire tribe. You can't really even put them to genus level oftentimes if you don't watch them closely enough. In that case, you can just write swift species. And if you're only writing scientific names, then you will call it Paorini. So it could be any kind of swift. It could be one of the branded swifts, or it could be rice swift or something else. So I have made a note of that. One, Urema. Which Urema? Okay. Try to have a look if you have the binoculars. Just try to see which Urema it is. Urema is grass yellows. Grass yellows are fairly easy to identify. You just need to make sure that you have looked at the identification features properly. So here we butterflies, grass yellows that will fly this far, this high up in trees. You largely have three spot grass yellow and common grass yellow. Small grass yellows and spotless grass yellows won't fly this high. So it's one of those two. So you just have to look at the underside of forewing to see whether it has three spots or two spots. It has come down now. We'll keep an eye, and when we have the identification, then we will change it from grass yellow species to yellow, grass yellow. Yeah. So I'm going to write this down. Grass species. Okay. As a note at the bottom of this 30-minute count, I'm also writing that the Jezebels were feeding on this particular cassia, and we'll identify the species level later. So I've written that down, and you can also write down if you see anything on a particular flowering plant. So typically, you should keep on taking all sorts of notes like this, where you can get more information rather than just numbers. If you're just talking about numbers, then it's going to be a little bit of a problem because problem, or rather, it's a limitation because you're not getting enough information out of all the information that you could potentially get. Why don't you come here so that you won't be against the light? So, for example, on this walk, while you're counting butterflies, if you can also note down butterfly and nectar plant associations, then that will be a good record, for example. If you see anything laying eggs, note that down as well. So it won't become part of the 30-minute count as such. There's a castor there. So can you check whether it's common castor or angle castor? So these guys will have a look at it, confirm it. So you can also write down these other notes, which will just enrich our understanding of what butterflies are doing in this particular area. If you see a if you see a congregation associated with butterfly migrations, then you could also write that down. Yeah. So typically, hold on there. Typically, what you can do is record all sorts of information. For example, if the migration is going on and you're seeing, let's say, 100 butterflies. So if you just say blue tiger, 100 individuals, or dark blue tiger, 200 individuals, then you don't know exactly, or people who don't know that most of these congregations are associated with the migrations, they won't really know. So you can also say dark blue tigers, 200 individuals, blue tigers, 100 individuals, part of the migratory congregation. Yeah. So write all these things down. There are two more lemon immigrants in the field there. I'm going to add lemon immigrant. Earlier we had seen only one. Now we have three.
And another booster.
Common
Common
castor.
castor.
Which one? Common castor. So we have got one common castor. Dark brand bush brown. I'm going to add one more to what we had earlier. Okay, let's go then. Keep going through now. Good. Okay, so there's one more Delius. Tarika had just noticed this, and it's feeding on lantana flowers. So I'm going to write down. Oh, that's a beautiful specimen, and people can now see it closely. Great. Okay, so that's our third confirmed individual, and I'm also going to write down that this particular one is feeding on lantana. Okay. We're also picking up nectar records, the nectar plant records as we go along. Okay, so that's it. What yes, this is a dark cerulean. You can take a close up. Do you see it right here? Yeah. Arko, you can try to go a little bit closer so that people can see the butterfly very well. So that's a dark cerulean. So that's a new species for this 30-minute count. So if you want, you can also note down that it's a male. So all these other notes, whether it's feeding on Lavlos plant, it's doing something else, these things will go under notes for that 30-minute count. So you can also continue to take this other information. And some of this can be useful in the long term. Some of it can act. Hmm? Another bush brown. Yes. Okay, this is also a dark brand bush brown. So I'm going to make that three now because we have seen three earlier. So Namrata is online, right? Can you ask her whether everybody can hear everything all right, so that we are sure people can get the whole experience and that they can hear us. Namrata, can you hear us?
Yes, I can hear you guys. I'm just typing
Good,
it
good,
to ask
good, good.
everybody's.
All right, great. All right, let's keep walking then.
We are able to hear you, but the voice is breaking, you know.
Once in a while breaking. Okay, that may be because of the internet access, but hopefully everything will work out. There's a nice teak tree here, quite a lot of flowering and fruiting, and we'll see if there are any butterflies feeding on these flowers. Very rarely I've seen some butterflies feeding on teak flowers. It's not a big butterfly plant, nectar plant, but we'll hang around here and see if there's anything else. There's more lantanas, so potentially there'll be more butterflies feeding. Unfortunately, because it's so clouded today, not a lot of butterfly activity, but otherwise at this time, we do expect other, uh, other skippers to be around. This flying? Dilemma, okay. Since it's flying low, let's try to see which species it is. You see? Okay, all right. Let's keep walking. Here? Okay. Yeah. All right. So that's also a small grass yellow. So I'm going to add that. Okay. Which immigrant? Lemon. Oh, that's a lemon. Another castor. No, this is a leopard. This is the one that we are talking about. This is Flacosia. Sarko, can you come here? This is Flacosia. This is the Labulos plant of leopards here. These leaves are a little bit older. They're not very fresh and tender. So most likely leopards are not breeding on this particular plant right now, although the new leaves are just beginning to come out. I think they might be laying eggs on this. As soon as these, oh, okay, I do see an egg there. There was a polytus, stitches. All right, I will write that down. So there's a very tiny egg here. You see this? Yes. Small yellow one? Yeah. So they've already started laying eggs. So we will see exactly how big this one is going to get, how quickly. But all these leaves hopefully will. Laying eggs there. What is laying egg? Ah, yeah. There are some eggs. Ah, okay. Yes. So this is going to be most likely common grass yellow. It is common grass.
There's lots of eggs here. 2, 3, 4, 5. Sometimes.
5 eggs, you said?
Yes, yes,
Okay.
5 eggs.
Lemon pansy. Okay. So that's a common grass yellow. Now you can see it's opening up a little bit and it's also becoming a little bit warm. So we have started seeing more butterflies. So this is lemon pansy that Arco is taking a video of right now. And there's a grass yellow flying right around it. And that grass yellow, see, Okay, that is also a common grass hello. I'm going to add that this is different from the previous one. This is much more fresh. It seems to have come out just this morning, whereas the previous one was a little bit older. I could see a little bit more wear and tear on it. So I'm going to write two common grass yellows now. Okay, let's continue.
There's another jersey.
Ah, good. Now there's sufficient distance between the previous patch and this patch, and assuming that these things are not following us, flying around with us, and these are the same ones that we're seeing. But again, you can look at the wear and tear on this butterfly and confirm that it's true. But as I said, there's a difference between the previous patch and that visible that we saw in here. So I'm going to assume that it is a different. Okay. So now our species list is rising because the sun has come up. Oh, I actually forgot to write one more. You had said we there was a common Mormon earlier.
Yeah, stitches.
Yes, female form. I'm going to say female form stickiest, and that note will go in the. Maybe I thought you want to come closer because of the wind. They may be not able to hear me that well. So it's a female form stickiest. So I'm going to note that separately, and that female form will go in the notes. Common Mormon will stay in the main count data. But later on, if you want to look at, okay, good. Genitia or Chrysippus? Chrysippus, okay. All right. So anyway,
Baron.
later on,
Yeah, that was a Baron female.
Baron, okay.
Just flew away.
Butterfly activity is really picking up. So there are two grass pillows there together. Some the common grass pillow from 2 to 3. Okay. All right. Fly. So when you're counting, among the things that you should be looking for is this kind of interesting biology. For example, nectar plant associations is one kind of information that can be useful in understanding not just which species are occurring where, but also what they're feeding on, what are their requirements to survive and thrive in a particular habitat. So that's one kind of biology that we can note down. Another thing we can do is of course, Lavalose plants that we have talked about. Third thing, for example, of interest today is that we are seeing some toxic species, some aposematic species, and of course, some mimics. So if you want to look at model and mimic ratios in the long term, this is another thing that we can do from the count data. If we have precise numbers of how many toxic species, models, and the mimics that we see, then we can analyze those patterns as well. And that's actually something that Viraj is doing right now from the Bangalore butterfly data. So slightly over 8.5 years of data. No, you have 9 years of data that you are analyzing right now. So our 10th year is on, but he has the 9 year data, which is analyzing. So from that, we will be able to do a very nice time series of models and mimics to understand exactly what their population dynamics are like. And then we can also get fairly unique insights into how mimics might be benefiting from abundance of models. There's one idea that models must be much more common compared to mimics for mimics to be effective. So if that is true, this is exactly the sort of data that we need. So there's a bunch of things that we can understand from this kind of data. So just keep recording as many details as you can. If there are sexually dimorphic species, also note down male and female for every single individual that you see in that count, so that we may be able to even get sex ratio data in the field from the butterflies that we observe. And then there are lots of interesting things. Most of the butterflies you see for a lot of species, for example, pale Jezebel, considered to be a very rare species. If you go to their habitat in the right season, June, July, sometimes you might see thousands of them in some areas, which is what we saw in Eagle Nest area. And we got really beautiful pictures. Probably the best pictures for the first time. This was back in 2015. We did not see a single female. Yeah, only males. These more than 1,000 individuals are only males. So this is really interesting from the biology perspective for the species. So you should really note down this kind of very striking things as well. And of course, if you don't know how to distinguish between male and female, then you just write the species name and the number of individuals, and that's good enough. But if you can get more information, keep on writing all these notes. This will make you a better naturalist, and it will generate a lot of data that will be useful, not just for your career, for your lifetime, but perhaps beyond your lifetime. So this is going to be really, really important going forward. As I mentioned, we don't have too much data in India, which is helpful both in research as well as in conservation. So if we keep on collecting this kind of data, I think it's going to go a long way. Okay, so Arko, can you turn around once again and let's get a 360 view of what kind of habitat we have around. Maybe walk a little bit so that you can get that gate and the fields around you. So while you're doing this, you will see that there are some buildings uh, coming up in the background. So if you keep on getting more and more buildings here, of course, GKVK campus will not change that much, but certainly the larger habitat area. If we do this for 10 years here with a changing landscape, potentially we should also look at whether that kind of development has had any impact here. And if the farming practices, experimental plots here change, then of course you're going to get a very different picture here than what you will get elsewhere. So if you're doing this in a sanctuary area, then perhaps the habitat is going to improve. Forest departments in all states, for example, are doing a lot to improve habitat for butterflies and other organisms, and not only larger animals. So if there are any improvements in the habitat for butterflies, then we'll also catch that. Viraj, okay. So that's our fourth one on this count. Yeah. And that was Pelopida species. So I'm just going to write it as Pelopida species. Sometimes, especially when butterfly activity is not great because of weather or something else, you could also try shaking some bushes, disturb some vegetation so that butterflies can be flushed. So here, there's a grass fellow sitting under this leaf. First, I'm going to check which species it is. I think it's going to sit down once again. Okay, this is a three-spot grass willow. I see three spots in its upper side, underside forewing. So you will be able to take it from that side. So this one is the first 3-spot grass willow we have seen on this count. But there are 2 now and 3. So there are 3-spot grass willows. So that is what you're going to add. So this grass is quite nice. We'll very likely find caterpillars of bush browns and evening browns and other things that feed on this. I also see this leaf has been eaten quite a lot, and actually quite a few leaves eaten here. So while I walk, guys, can you try to find caterpillars of whatever you can? Most likely, this leaf damage here that you see is from caterpillars. So just keep looking. All these caterpillars, bush browns, evening browns, rings, rings also feed on this grass. All the caterpillars will be on the underside. So keep turning the leaves and see if you see any caterpillars. Now they also drop easily. They know that the host plant is all around, so they will drop at the slightest hint of disturbance. So be careful. But if you find any caterpillars of bush plants, that will be nice. We'll get some good pictures. That those are eggs, but they're eggs of some other insect. Most likely a bug. We'll have to take a close-up picture or look at them under a microscope to find out. But at least from the shape, they look like eggs of some hemisphere. That's actually a very nice grasshopper. It's time now, 11:10. Oh, good. So it's closing off our first 30-minute counts. So I'm just going to write the next time slot, which is 11:10 to 11:40 a.m. And that's our first
That's
butterfly.
ring.
Of
That's
this.
the fourth.
This is going to be a common reading. So we'll put that in the next one. So this was right when we were starting a new count, but it is exactly 11 past 11 right now. So I'm going to put it in this count. That's going to be common reading. And for this particular count, what I'm also going to write is that more sunlight and then warmer. Okay. And of course, as we go around here, we'll take more videos. We'll look around so that we can see exactly what kind of weather it is. Marco, now we can come a little bit closer. So what we have done is we have just finished our 30-minute count and we have recorded those data. So just as a summary, I'll count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. We have seen 19 species of butterflies in this particular 30-minute count. Not bad. And especially considering that it was clouded earlier, 19 is really not bad. And the number of individuals I'll have to count, but it seems like we have seen at least 30 individuals because some of them we have seen 3 and 4 individuals, 5 individuals for lemon emigrants. So probably much more than 30, 34, 35 perhaps. So that's a typical 30-minute count, especially when the density of butterflies and the diversity is a little bit modest. If we were in, let's say, some nice evergreen forest uh, in the Western Ghats or in the Northeast or Andamans, then we'll probably have seen 40, 50, even 60 species in this half an hour, and potentially many more dozens of individuals, potentially even more than 100 individuals. Or in migratory season, we might see again a congregation of several hundred individuals. So this is what the count data are going to look like. And of course, we will have a tab delimited text file, CSV file, some kind of a data file, which you can organize in an Excel sheet as well, or open and organize it in Excel. So all this information is going to get recorded in a standard format in an Excel sheet. or a database really, and then we can analyze it in R or some other software where we can do this kind of analysis. But we'll probably have another session for that kind of analysis. Maybe Viraj will lead it at some point. So we'll do that another day. But why don't we actually get into the shade a little bit? Let's just sit down here, all of us, and then let's see if people have any questions about what we have just done. And especially, I know that a lot of people are taking 30-minute counts right now. In the past few days, I know that people have started taking counts. So if you have taken counts and if you're going to have any questions, you can take those questions. Note that we have just walked on this path and we are going to go back there because our vehicle is parked there. But on the way back, we are not going to take 30-minute counts. If you see any species that we haven't seen today, we'll just note those down, but we won't take another 30-minute count because we might be counting the same species, same individuals, I mean. And therefore, there's no point in taking a count on the way back because that's pseudo replication, as I mentioned yesterday. All right, so that's a practical of 30-minute counts. And let's see if anybody has questions. Namrata, can you please check at the chat box and see if there are any questions?
Yeah, we have quite a few. So Prerna has been asking, like, how do we count active butterflies in a congregation?
So we covered that in the talk yesterday as well, but I will answer this because this is an important question. So if a congregation is large, beyond, let's say, 10, 15 butterflies, 20 butterflies, if they're sitting down, you could still count them reasonably well, even if the group is a little bit larger. But if they're moving around actively, then there's no way you're going to count them precisely. So there you can write down your best estimate. So if you think that there are about 20 butterflies, give or take 5 plus minus, then you can just mention that. If you think there are 50 to 100 butterflies, then write that. And when we are analyzing data, we'll take the lower margin, 50, as the number. But basically make your best estimate and note it down. There's no way to do this. If you want to do this kind of work, then the only thing that you can do, and this is really important for studies in population biology, if you're going to do a very intensive survey of a particular endangered species, or legally protected species or endemic species or any species that you're interested in, such as, for example, we have done some work on Bhutan glory before, the population biology of Bhutan glory in Arunachal Pradesh. So for this kind of projects, what you can do is you can mark and recapture. So that idea is, and again, for this, you will need a permit from Forest Department or wherever else you're doing your research, because this is a slightly more involved method. So it's best if you get permission to do this. But what you could do then is catch every single individual of that species that you can find in the area in which you're doing the study, and then write a number on the butterfly with a marker, and or you can use stat. There are various ways to do this. We will have perhaps a separate session on this at some point, but you can basically individually mark that butterfly and then release it, and then keep catching butterflies. So if you do hundreds of butterflies, if you mark hundreds of butterflies, then you are going to get some recaptures, meaning that you will recapture an individual which you had marked distinctly earlier, and then you've got it. And my PhD student Vaishali Bhaumik, for example, has done that kind of work on the migratory denines. So for dark blue tiger, and then crows, common crow and double-banded crow. These are the 4 species which predominantly migrate in southern India in response to the monsoonal cycles that we have. So what Vaishali did was she marked all these individuals, and we had recaptures up to, I think, 30 or 40 days beyond initial capture and marking. So when you do this, you can then use a simple calculation. If you have marked, let's say, 100 butterflies in that particular season, and you were able to recapture 20. So if your recapture rate is 20 and you have caught 100 butterflies, you can then calculate exactly how many total number of individuals there could be. Of course, it gets more complicated if your mark recapture goes on beyond, let's say, a month or so, which is the time when butterflies can potentially breed and produce the next generation. Then it messes it a little bit. But within the same generation, if you do intensive capture recapture, then you could estimate population. So in that case, you could get slightly more reliable estimates of how many individuals there are. And you could do this for congregating species as well. But again, that's more involved. If you don't want to do this, just estimate how many individuals there were. Okay. All right. Next question.
So Karma Jamsho has a question like, is there any app where we can submit the count data? Like eBird is there.
Yes. So just hold on for a couple of months. We are going to release our mobile app soon. It's being actively developed and it's developing really, really well. It's going to be beautiful. You'll be able to submit all kinds of data in a standardized format. And the format is, I think, a lot better than what most other apps provide. So just hold on. Until then, you can just take notes on your mobile phone or on your in your notebook. And then right now we are compiling all that information for BBM. But eventually we will have the mobile app, our own mobile app, which will serve us a lot better because we'll have features that are not easy to find in other mobile apps. So just hold on. Hopefully by November or early December, we might have that out. So after that, you can make this your New Year project to a a big counting effort using our mobile app.
But now you can submit the photo in iPhone butterfly.
Photos that you take, all those pictures can be submitted to iPhoneButterflies.org or Butterflies of India website. So there, for example, if there are a bunch of people in the group, you could be taking pictures and you could all submit them to Indian Foundation for Butterflies website. And that's a very good database. We have 1,14,000 observations as of yesterday. So it's a very, very good peer-reviewed database. If you want to enter data there, it's fairly easy to do it. So you could just submit all the observations there for now. And the exact count data that, as I mentioned, you can submit through the mobile app when it becomes available in a few months. All right. What's the next question?
So Reshma has been asking like, what's the correct time for taking a count and how will she be able to differentiate between male and female butterflies in the field?
Okay. Remind me about the male and female butterflies later, how to sex butterflies. Regarding when to do your counts, in most parts of India, I would recommend from about 8 or 9 to about 1 o'clock. If you're going to do this on a regular basis, if you're going to do this, let's say every alternate weekend in a particular area, like we have been doing in several parts of Bangalore, then what you should do is do it from 9 to 1. It's fairly convenient. If you are in a city or town, if you're doing this in whatever natural habitats near you or in an urban park, then I think by 9 o'clock, there's enough light, there's enough heat that butterflies are going to be quite active. And 1 o'clock, lunchtime. So you can just conveniently do this from 9 to 1. If you're really crazy about butterflies, if you really want to do this, what I would definitely recommend you is that in your particular area, you can take counts for a longer duration. And there, again, you can decide exactly where you're doing your counts and what would be the best time. If you're in an evergreen forest with really tall trees, for example, you're not going to get enough heat inside the forest before probably 8:30 or 9. So there's no point in starting your count a lot earlier. If you're in a grassland which heats up really fast, and especially if you're doing this in, let's say, spring or summer, in India, temperature is going to rise really, really quickly. So you better start at perhaps 7 or 7:30 and do it for only 2-3 hours, because beyond that, the sun is going to be too intense for most normal people to continue doing your counts. So in that case, you could do counts only for 2 hours or perhaps even 1 hour, because even butterflies might get overheated if the temperature is really high during the summers, and they themselves might be active for shorter duration than, let's say, in the same habitat during the monsoon. or when it's otherwise not so hot. So check what is best in your area. The general rule of thumb is in most areas, and also considering your convenience, I think 9 to 1 is what I recommend. But you can tailor it to your area, to your needs, to your goals of why you're doing 30-minute counts. But once you decide that you're going to do it at a certain time, if you're going to do it for seasonal occurrence of butterflies, then definitely stick to the same time window so that your data can be compared. You can't compare data, your 30-minute count taken from 8 to 8:30 with your count data from 1 to 1:30. So fix the time, fix the area, and then keep doing it uh, every alternate weekend. Yeah, so that's a general rule of thumb. Oh, and the second question was, how do you distinguish between male and female? We should probably have a separate session on this sometime. But many species are sexually dimorphic. You can very easily identify male and female of Denaidic fly or Greatic fly or common Mormon mimetic female forms and males, for example. So when you can easily identify males and females, you can note those down. If you get really good, then you can identify male and female even from very subtle differences such as wing shape. So females typically have slightly broader wings compared to males. And it's not true that you can distinguish between male and female in every single species this way. For example, in pansies, you can't differentiate between male and female based on wing shape, but you can differentiate male and female in, let's say, ape fly or common rose, crimson rose, that kind of butterflies. So you can have differences in wing coloration by which you can tell male and female apart really easily. You can tell male and female apart by wing shape. You can tell them apart by abdomen and various things. So there's no one rule by which you can differentiate between male and female, but use all the information there is. Unfortunately, not many field guides give this kind of information, but some field guides do, and there you can get this information from which you can learn to identify male and female. Next month, I'm finishing another book, and that's going to be really, really useful. You will have this kind of male-female differences, seasonal differences, and other kinds of variations described in this book. So that will be really useful for you to learn how to differentiate between male and female or seasonal forms, and also differentiate that from individual variation that some species show. Yellow orange tip, for example, there are seasonal differences and there are certainly sexual differences in the species, but the individual differences themselves are also pretty striking. So if you have seen lots and lots of butterflies, lots and lots of individuals of the same species over seasons, over years in different areas, then you will be able to slowly understand how to distinguish between seasonal variation, sexual variation, and individual variations. And since I've been doing this for a long time, I have this kind of information which I'll be putting in this book. So again, wait for a few more months and you will have this field guide released. It's not going to be expensive and it is going to have a lot of material in it. So if you're looking for a field guide, just wait for a little bit, and I think you'll be able to use it for all kinds of information on biology of butterflies, plus identification, including citizen science kind of projects. So just hang on. Until then, of course, you can go to Butterflies of India, and on Butterflies of India, we have already identified male and female wherever they can be distinguished. So you can go to our species pages and then just see what are given as male and female, and you can see what are the differences there and learn it that way. You could also join our Facebook group, Butterflies of India, or WhatsApp group, Butterfly India and Butterflies of India. There are two groups. So you could join those groups and then see, you can post images or you can just ask questions, and then other people will help you with this information. There are lots of experienced people. There's Ashok Sengupta, Rohit Girotra is there, Pranav Gokhale is there. Who else is there? Sarika is there, Viraj is there, a bunch of other, oh, Hemant is there. So there are lots and lots of people who are there. You could get help from them in understanding how to differentiate between all these different species and sexes. Yeah. What's the next question?
So next we have two requests for you. So people want you to take an introductory session on the basics of butterflies, like cover these things in more details, which we are discussing about how to differentiate between male, female, and things like that. And secondly, they really want you to take a session on analysis of butterfly data.
Okay. So for butterfly data, let's see. What I will recommend to people is, if you have data, send me maybe 3-4 line description of what kind of data you have. What will be really good is we wait until, let's say, another 6 months or 8 months. Let's see what kind of data people are generating. Today's session was on 30-minute counts, but there are lots of other ways of collecting data. And I've mentioned some of these things in today's session, as well as in other sessions that we have done last year and earlier this year. So for example, I mentioned you could also do capture, mark, recapture. You could also just be focusing on data on nectar plant, and Sarika and some other people have actually taken a lead in putting together a data set on nectar plant use. Uh, Sarika and group has also done an experiment on effect of pollution on survival of the common castor. So you could do various kinds of projects. Counting butterflies is only one of them. It's an important activity, but there are many other ways of doing this. So in the next 6 months or so, if you have a project in mind or you have collected data, write me 3 to 4 lines of what you're doing. I will compile that, and then we'll decide what kind of analysis we can demonstrate, right? And my PhD students also have a lot of experience now of analyzing this kind of data. Vaishali is not here today, but she has a lot of experience of analyzing data in R. So maybe she can do a session on analysis in R, especially with respect to butterfly data. And she has experience analyzing morphometric data and um, migration related data. So we can gather a bunch of students around. Vijay knows how to analyze some of the kind of data from iNaturalist and several other places. So we'll gather a team of researchers who can have this session on data analysis, and perhaps we can do a series on various kinds of analysis. So please send in your queries, and then we'll organize it. Yeah. Instead of doing a session on what we do and the kind of data analysis we do, let's do it with your contributions first and then analysis. Okay. What's the next question?
So that's about it, unless anybody else wants to ask something to Krushnamegh.
Let's wait for 2-3 minutes. And
Yes,
if more,
I have a question. Hi,
yes.
Krushnamegh, this is Ashwin.
Hi, Ashwin.
You know, in Nanda Devi in 1993 in the Inner Sanctuary, when I was completely clueless about this, I was two biologists, showed me a method to take spot counts and do some kind of simple, butterfly survey there.
Okay.
Based on some discussion that we had, in the evenings after, when it was dark, they told me that because the terrain was extremely dangerous and it wasn't wise for any of us to go anywhere else alone, and every one of us had our tasks. So they suggested to you pick a safe spot, sit there for about 20 minutes, and whatever comes within as close you can record, which was approximately 20 meters or so, and note these things down. So if we had done it for 20 minutes, but would that be a valid? The people were Hari Shankaran and Satyakumar. So
WI.
yeah, yeah, but Hari Shankaran was SACON late. So that data, would it be meaningful?
No, it will be meaningful. So the thing about doing any kind of scientific work is to pick a method and then consistently follow it. So what we are doing is the 30-minute count is because if you sit in one place, you are going to see relatively fewer butterflies. And then you're also not going to capture a lot of variation. Whereas if you keep walking, the chances of you seeing more butterflies and then capturing greater diversity are just higher. Also, if you're going to do, let's say, surveys of butterfly diversity, and not only seasonal counts, so that so seasonal counts will be, you pick up one or two places and then you count butterflies every alternate week there, so that you get to know how the population cycles happen in these butterflies, how the population numbers go up and down. That's one reason why you want to do 30-minute counts. Another reason is you can get a fairly good list along with quantitative data on butterflies there. So if you're doing surveys in an area, let's say to help forest department come up with a list of butterfly fauna with some data on populations, then you would do it in multiple places. You may not go to the same place again and again every alternate week, but you will go to different places. Each spot you could do maybe a few times, but not every alternate week. So you will be able to get a lot of data that way. But there may be cases where you really cannot walk. For example, as you mentioned in Nanda Devi, since it was dangerous, you could not really walk around too much. Another really genuine case where you cannot reasonably hope to walk or move about is, for example, when you are taking counts in the canopy. So there you're going to use some kind of climbing equipment. You go and sit inside in the tree, and then you're stuck in one spot, right? Unless you do a canopy walk, there is no way to do this, and nobody can do canopy walk in India right now. So then you are sitting in a tree, ideally a slightly taller tree than rest of the trees, so that you can look at the crowns of these trees from a comfortable branch in this taller tree. And then you have a certain circumference around you where you can count butterflies. And I've done this kind of work in the Animalize actually way back in 98, 99 doing my master's work. So I do have some count data. So this method is called point counts. Okay. So transect method or 30-minute method is you have to cover a certain distance. Point count is you sit in one place and then decide on a circumference or rather radius around you in which you will count all the butterflies. So 20 meters is a perfectly good area. And if you could still walk around in those 20 meters, you could do that, or you could strictly sit in one spot and then just see what is around you. Okay. So that method is perfectly legitimate. People have been doing this for a long time. And in fact, I learned of the point count method in WI, and Satyakumar was one of the teachers who taught us this. There are some more advantages of this. For example, for plants, and this is something that Rawat sir from WI taught us. You could go to any area and then just say every 2 minutes or every 5 minutes, you're going to stop. And in the 10 meters around you, you're going to, let's say, count trees of 10 centimeter girth or larger, for example, right? So you could also do this kind of a thing where from a point you have 10-meter radius, and in that 10-meter radius, so you know the area, and then you count every single tree in this area, which are 10 centimeters and greater. Or we could do it for butterflies, or we could do it for something else. And you could also calculate the distance between you and the organism, whether it's a tree, it's a butterfly, it's anything else. But then this is a standardized method that now you're going to use multiple times. So if in some places it's dangerous to walk, impossible to walk, point count method is perfectly legitimate. For butterflies, under most circumstances in India, that really won't work as well as your 30-minute counts. And therefore, for butterflies, I'll recommend 30-minute counts as a standard procedure. But point counts are perfectly legitimate. Nothing wrong in it. If you want to stick to that method for certain areas, for a certain project, absolutely do it.
I'd like to just add one more facet.
Data are not bad. They're not unusable. You will just so depending on which method you use for counting, you're also going to use a different method for analysis. So know that as well. But many ecologists in India will be able to help you analyze that kind of data. There are very standard methods for analyzing this kind of data. So just look around, or you can contact me and I can put you in touch with people who can help you analyze those data. So anything is fine as long as you do it consistently, is basically the lesson you should learn. And 30-minute counts is what we are now we have been using for 12, 13 years. So we know that it works really well. So you could go ahead with it. If we stick to so advantage of doing this, which is that all our data will be compatible. If somebody does 1 hour count, if somebody does 3 hour counts, then the power to compare becomes, it diminishes pretty fast. If we stick to one method, then it's much easier to do this. And therefore, I'll recommend that you do this. Okay. Any other questions?
I just wanted to add one more facet. That
Okay.
is that we use the spot count on their advice. They pointed out various, terrain altitudes or biomes or different kinds of stands of forest. And so all along our, exfiltration route that is coming out from the sanctuary, we stopped at evergreen forest or at coniferous forest or at dwarf juniper and saw the various kind of this thing over an altitudinal this thing. So the spot method that we used it in this way to get some kind of an idea of the way the butterflies are distributed in that park.
Sure. No, that's great. Especially if you did this altitudinal and habitat gradient, then actually those data sets will be really interesting because you could analyze patterns of occurrence in 92, 93 when you did this. And I don't know whether you have written up anything on that, but you should probably analyze those data and publish a paper at some point because that data set, as long as you have captured all the species at that time in your count data, you should be able to write a pretty good paper on that. So please do it because if you have done work, but if you don't publish or if you don't make the data available, then it's basically lost to the rest of the community, right? So definitely please do analyze the data, write up a paper, and then make data available to the community.
It's in a report of the expedition, and the only peer review was by these two gentlemen. So but it's not in a peer-reviewed journal as such.
That's fine. You could still write up a paper and submit it. So if you have the report, you could send me a PDF file. I could have a look and perhaps advise you on how best you could analyze data and what kind of journal in which you could publish this.
Fine, I'll do that.
Great.
Thank you.
Any other questions?
So Jyotika has been asking, like, should we count any early stages we encounter?
That's a very good question. So the 30-minute count that we just did is a general method which can be used for adult butterflies, especially those which are active during the time when most people want to go out and watch butterflies. But there were also specific projects for which you could collect data. So what you said, early stages, there's a different method that you will have to employ. The 30-minute counts is not really an appropriate method because unless you mark out a trail where you have marked your larvalose plants, there is no point in doing this. And if you have done this, even there, 30-minute counts won't be appropriate. There, what you should really do is you identify an individual plant and then keep going to the same plant over 2 or 3 years, and then see what are the seasonal dynamics of use of that particular host plant. And of course, you shouldn't do just one individual of that host plant. You should ideally choose at least 10 or 15 or 20 plants, individual plants of that particular species, and then monitor incidence of early stages of the butterflies that you know, or moths or any other insect that you're interested in. And then take down data for that particular individual. You can mark those plants as plant 1 to 15. And then you could write down in January on this plant how many young leaves were there, how many fresh leaves were there, and then whether butterflies were breeding or not. If there were caterpillars and pupae or eggs, how many did you find? Yeah. So you could devise your own method for the kind of work that you want to do. If you want to be more rigorous with this, what you could also do is you can identify 15 plants where you will take these data. You can identify another 15 plants right next to the plants that you are counting. And from those, you can take leaves, a certain number of leaves every month, let's say 10 leaves. And you could do some kind of analysis of how fresh they are, whether they have high water content, low water content, high metabolic content, low metabolic content. That will give you nutritious value of leaves. And then you could also take climatic data along with host plant data, and then you could see how well butterflies are breeding through these different months. So there are really different ways of doing things based on what project you could do. So if you want to do this kind of work, don't worry about 30-minute counts. That is not going to be appropriate. But you could identify host plants and then do this kind of a project. What may be useful though is you conduct 30-minute counts along with this work on larval host plants and caterpillars, so that you will have some sense of what the density of adult butterflies flying around was. And then you could study larval stages. And that will actually be a fantastic project, because then you can look at population biology of the butterflies of a particular species that you're looking at a lot better. And that's the kind of work that I've really been meaning to do for quite some time. So if any of you are interested in doing this, talk to me. I can advise you on how to do this. If you're going to be a PhD student or a master's student, I'll also be happy to advise you. You could try to join my lab. Prepare well, read well, become, if you have motivation, become a little bit better trained as well. Come to my lab for master's or PhD project, and you could also do this. So your specific question, your specific method is a short answer to what you just asked, but you could do larval host plants and caterpillar interactions or breeding biology of a species as well. And the methods will be different. Okay, what time is it? We probably don't want to keep the recording for way too long. We started at 10:30. So actually, it's only been 1 hour 15 minutes. So maybe let's say we are going to do this for 5 more minutes max, or 2 more questions.
Yeah.
Yeah. So are there any other questions?
So Reshma wants to know whether there's a butterfly walk organized in Bangalore and how she can join it.
Absolutely. Viraj, why don't you talk about it since you go there regularly?
Yeah, so there is a Bangalore Butterfly Club, which do a walk every 2 weeks. But currently, due to pandemic situation, we are limited to 6 people. So I guess in some months it will open, and after that, like anyone can join.
So after that, if 20 people come, 30 people come in a particular weekend, not a problem. Sometimes you have groups. I think the largest groups you have had are close to 30 people. So group size is not a limitation. If the group is too large, we will split into multiple small groups, each of 2 to 4 people, 5 people, so that we are not too crowded, because everybody should get to see butterflies properly. So in that case, we'll split into smaller groups. But as Viraj mentioned, right now, because of the pandemic, there is a limit that Forest Department has imposed, saying that you can't be a larger than 6 people group. So right now, there's an issue, but maybe Viraj can contact Viraj, or you can drop me an email, Krushnamegh@ifoundbutterflies.org, and I'll put you in touch with Viraj and Nagesh and Rohit and other people who do these counts on a regular basis, and you can join the group. In fact, I will urge you to join the group, and you should really learn how to watch butterflies, how to do 30-minute counts with this group, and ideally you should pick up another place nearby you where you can start your own 30-minute counts. Ideally, that is what we should be doing. Do it in as many places as possible. So we'll try to organize that. Okay, what's the next question? Nothing? Great. All right. So let's stop then. It's been a nice hour and 15 minutes. And again, it's a pleasure to talk to all of you about this particular method that we have been promoting and using for quite many years. And I hope that our network will grow and that people all over India will start doing this sort of counts, so that we go from serious butterfly photographers and watchers to citizen scientists. And perhaps even some of you scientists in the long term, especially if you're young students. I wish you best of luck. Stay in touch and drop me an email if you have any questions. And certainly, I would like to hear from you, and I'll be very pleased if you become part of our network. Yeah. Bye. All right.
Thank you, Krushnamegh.
Okay. Bye.
Bye.
Stop recording.